Okay, sir. Now I can.
Let me know when it's visible. Is it visible to you, Atif?
Yes, sir. Now it's visible.
Okay, very good. Everyone, is it visible?
Yes, sir.
Yes. So these are some questions from this chapter that have come in previous years, and I have some interesting ones marked. Okay, let's start with question number 8 because we have studied amniocentesis, right? Let's start with Hanifa. Hanifa, are there? Can you speak? Will you please read the question and tell me the answer? Others can read the question and give me the answer in the chat, just to me. Yes, Hanifa.
In context to amniocentesis, which of the following statements is incorrect? A, it can be used for detection of Down syndrome. B, it can be used for detection of cleft palate. C, it is usually done when a woman is between 14 to 16 weeks pregnant. It is used for prenatal sex determination.
Yes. What do you think is the right option?
It can be used for detection of cleft palate.
Do you think option B is incorrect? It's asking incorrect. Which is incorrect? Which of the following do you think is incorrect? I received Rifat and Atif's answer. I'll be discussing the right option. Okay. Hanifa, what do you think? Which one do you think is the right answer and the wrong, incorrect statement? If you're speaking, you're not audible to me.
I'm not sure, sir.
Okay, let's go statement wise. Okay. Do you think statement A is correct or incorrect? Can amniocentesis be used for the detection of Down syndrome, according to you?
Yes.
Yes, because Down syndrome is a chromosomal abnormality, right?
Yes.
Yes. So it is a correct option. So it cannot be the incorrect statement or the right answer, right? B,
Yes.
it can be used for detection of cleft palate. You know what is a cleft palate? No? You know or you don't know? Okay, so let's come to option C. It is usually done when a woman is between 14 to 16 weeks pregnant. Is this correct? 14 weeks means how many months? More than 3 months, right?
Yes.
Yes. So yes, it is usually done after 3 months, and you know the reason why it is not done before 3 months, because it can be misused, right?
Yes.
It's not, the statement is not telling it can be done. It can only be done. It is saying it's usually done. So that's correct because of government guidelines. It's usually done in this period. D, it is used for prenatal sex determination. Yes, right? It is. So option A, C, and D are correct. You know, so which is incorrect? The only left option B has to be incorrect, but let's understand why it is incorrect. So Atif and Rifat has also given me the right answer. Hiba, you are not giving answers in the chat. You have to write answers, Hiba. Atif, what is a cleft palate?
Not sure, sir.
You also eliminated the rest 3 and thought that B is the only one.
Yes, sir.
Okay. Elimination sometimes work, but you should know cleft palate. Remember we talked about the upper lip at the time of sometimes uh, a baby is born with an abnormality where the upper lip is not joined properly. It has, it has a cleft, like it has a furrow. Remember? Palate is that upper roof of your buccal cavity. So if there is a cleft in that palate, or if this lip is not joined, these kind of abnormalities cannot be detected through amniocentesis, okay, because they are physical abnormalities. Is it clear? Cleft palate can be detected by a very simple technique called ultrasound, because that is through ultrasound we see the structure of the fetus, right? Okay, everyone?
Yes.
Yes, sir.
Yes, so the right answer is option B. Okay, so yesterday if you did gift, so let's start with question 9 and I'll go to Rifat. Rifat, right there?
Yes, sir.
Yes. Question 9.
It's it's D.
Can you read the question, please?
A childless couple can be assisted to have a child through a technique called GIFT. The full form of this technique is A. Gamete internal fertilization and transfer, germ cell internal fallopian transfer, gamete inseminated fallopian transfer, gamete intra-fallopian transfer.
Yes. Which one is true?
D.
Yes, D is true. Gamete intrafallopian transfer. You also studied ZIFT yesterday. What is
Yes.
ZIFT?
Zygote intrafallopian transfer.
Yes. So either you can put gamete, which is the sperm, or you have to put after fertilization zygote. Yes. Am I visible to everyone?
Yes.
Yes, sir.
Okay. Good. Atif,
Yes, sir.
like to have read question number 3.
Okay, sir. In case of a couple where the male is having a very low sperm count, which technique will be suitable for fertilization? Gamete intracytoplasmic fallopian transfer, artificial insemination, intracytoplasmic sperm injection, and intrauterine transfer.
Yes. Which do you think is the right answer?
Option B.
B. Okay,
Yes,
first
sir.
point, write down very low sperm count is also known as oligospermia. Did I tell you this?
No, sir.
Oligo, write down, because instead of very low sperm count, if they use the word oligospermia, it means the same thing. Write down oligo, O-L-I-G-O. Oligospermia is where the sperm count is very, very low. And azospermia, A-Z-O. Azospermia is no sperm, like where the sperm production is not there. Like sperm production is very, very happening. Am I audible, everyone? Atif?
Yes, sir.
So azospermia and oligospermia written. Now let's come to the question. So you're saying option B. Okay. Others? I'm not receiving answers. I've received for Rifat, Hanifa, and Hiba. What do you think? Hiba, are you there? Hiba. Hiba, am I audible?
Yes, sir.
Yes, Hiba, you're not giving answers in the chat. You're understanding the questions? Rifat, which one should I consider as an answer? You're giving me two options. Atif, you are saying B. Okay, let's go to option A. Gamete intracytoplasmic fallopian transfer. This is it's not anything. Okay. Rifat has changed the answer to C, and Atif is saying B, artificial insemination. Yes. So if a male has very low sperm counts, then you can do artificial insemination. Intracytoplasmic sperm injection, intrauterine transfer. If you do intrauterine transfer, it won't. Intrauterine transfer is done for the embryo, right?
Yes.
Intracytoplasmic sperm injection. This is basically IVF. C is just IVF. You take one sperm and you put that, you transfer that sperm directly into the ovum through a micro injection. So that's called intracytoplasmic sperm injection. So don't you think, Adif, C can also be done?
Yes, sir. I think, but yeah, in the textbook and all, it's specifically given that artificial insemination technique is used for a low sperm count. So I feel like that's the more probable option.
Yes. So both Atif and Rifat, you're correct. Artificial insemination also is the answer, and intracytoplasmic sperm injection can also be done. So I think in 2017 this question must have, sometimes if these kind of question come, if you choose B or if you choose C, then either this question is cancelled and everyone who has attempted is given the marks. Whoever has selected B or C is given the marks, because both B and C can be done. See, technically low sperm count is just due to low sperm count, there is very less chances of fertilization, right? So fertilization becomes very difficult, because almost all the sperms, because they are already low in number to begin with, they die in the female tract, right? And by the time they have to fertilize the ovum, no sperm is left alive. So to assist this problem, either you can do artificial insemination, in which more sperms can be collected from the male, in multiple ejaculations, and all of that can together be inseminated in the female. So that serves the purpose. Or you take one sperm from the male and one ovum from the female and do IVF, intracytoplasmic sperm injection. That also serves the purpose. Yes, C is more costly and more tricky, but B is simpler, but both can be done technically. So logically, B and C both are correct options. You understand? Everyone understands?
Yes.
Yeah. Okay. Others are easy. I'm just discussing with you some important ones. Others I know you will be able to do. For example, if we come to this one, which of the following is not a sexually transmitted disease? You can tell me which is not a sexually transmitted disease. Question number 10. Trichomoniasis, elephant encephalitis, syphilis, or AIDS. So you know AIDS and syphilis are there, right? Trichomoniasis also can be transmitted sexually. Write down. Encephalitis cannot transmit sexually. So encephalitis is not a sexually transmitted disease. Just write down this. Coming to 11 again, which of the following viruses is not transferred through semen of an infected male? Again, it is asking in another words, which of the following is not a sexually transmitting virus or a disease? So which one do you think? Chikungunya virus, Ebola virus, hepatitis B virus, or HIV? Is not transferred through semen specifically. So if it is not transferring through semen, it is not sexually transmitted, right? Hepatitis B and
Ebola
HIV.
virus?
Sorry? Yes?
Ebola virus?
Chikungunya. Simple, chikungunya every year, chikungunya, malaria. So malaria happens due to a protozoa, chikungunya happens due to a virus, and chikungunya virus is not it does not go, like it does not attack. Ebola can reach testes and it can be transferred through semen. Okay. Chikungunya can't.
Okay.
So you can just write down chikungunya cannot be transferred through semen. Rest. Important questions for. Yes, question number 26. Atif, will you please read?
Okay, so copper ions released from copper lacing intrauterine devices, IUDs, make uterus unsuitable for implantation, increase phagocytosis of sperms, suppress sperm motility, prevent ovulation.
Yes, so these are very like close options. So you have to look at copper ion, specifically what copper ion can do. Okay. So which one do you think can be the answer?
So is it C?
Yes, the answer is C. It cannot make uterus unsuitable for implantation. For that, we need some hormonal regulation as well, right? So hormonal IUDs can do that because they release hormones which make the uterus unstable, and then it cannot host the incoming embryo. Is it correct? Just copper
Yes.
ions cannot do that. Increase phagocytosis of sperms. Now what phagocytose the sperms? They are the immune cells or the white blood cells of the female. So copper ion again cannot do that. Prevent ovulation. Again, ovulation and uterus maintenance both are hormone related things. So for preventing ovulation also we need hormone like progesterone. If you keep giving progesterone, ovulation is delayed, right? Because the uterus feels like it is in a pseudo pregnancy state. So ovaries do not release ovum if the levels of progesterone is very high already. Remember in the cycle where we made the graph, the progesterone comes after ovulation from day 16 onwards in the secretory phase. Remember, everyone?
Yes.
Yes, that's why. So the only thing, viable thing is it can directly, because they are ions and they can change, they can contribute to the chemical balance of the semen. They can directly affect the sperm by suppressing their motility. Okay. Good. Very good. Let's go to one more question. It's a very interesting one. Yes, this one. Question number 31. Hanifa, are you there? Will you please read question number 31 for us?
Two opposite forces operate in the growth and development of every population. One of them is related to the ability to produce at a given rate. The force opposite to it is called.
So this is not like directly and very clearly given in NCERT, but let us understand this question. The question is saying two opposite forces operate in the growth and development of every population. One of them is related to the ability to reproduce at a given rate. Remember, which is called the birth rate. We did that these concepts, but the question is just twisting the same thing and questioning you. So one is ability to produce at a given rate, which is like the birth rate, right?
Yes, sir.
Yes. So, but the question is asking that both these two opposite forces operate in the growth and development of every population. So if one is related to the ability to reproduce at a given rate, the force opposite to it will be what? Fecundity, biotic control, mortality, environmental resistance.
Mortality.
Again, is it just mortality that can restrict a population? See, it is not directly asking you birth rate. Then for that it would have been mortality. It's saying one of them is related to the ability to reproduce at a given rate. Now ability to reproduce at a given rate also can be affected from things apart from birth rate. For example, if something is opposite to the ability to reproduce, then does it only have to be mortality? Can't it be infertility that you just studied in the last class? It can be infertility, right?
Yes.
Yes. Mortality means the population is dying, but infertility is when the population is not growing at all. Whatever it is stable, and then everything dies and then collapses. And this is how the species go extinct. So fecundity could not be the answer. Biotic control is not clear. What kind of biotic control? Yes, it is some kind of a control. Infertility is there, but what biotic control is not clear. Mortality is directly opposite to birth rate, or what do you call opposite of mortality? Virality. Environmental resistances includes, I think, everything. The drugs, infertility, other issues due to which temperature, because in many species, temperature also plays an important role in both sex determination and birth of the babies. For example, uh, if we talk about every population, about chicken. Have you seen a chicken sitting over its eggs? Birds sit over their eggs to keep them warm. Why do they do that? Any idea, people? Why do birds sit on their own eggs?
Keep them warm.
Yeah, because that factor, environmental factor, is very important for their proper growth and development, right? Even if a bird lay eggs, but it doesn't, the environmental factors are not proper, the population will not grow. Okay, it will be opposite to the growth and development of of that population. So the most apt answer is B. B is not completely incorrect. Even biotic control is not completely incorrect, but sometimes I told you in biology we have to look for the best answer, the first best answer, then the second best answer. The first best answer is environmental resistance, which includes everything, which is opposite to the force which helps in growth and development. You understand this question along with this one? Where is it? Yes, 24. Rifat, will you read 24 quickly?
In vitro fertilization is a technique that involves transfer of which one of the following into the fallopian tube? A. Embryo only up to 8th cell stage. B. Either zygote or early embryo up to 8th cell stage. C. Embryo of 32 cell stage. D. Zygote only.
Yes, think about it and give me the answer. IVF, in vitro fertilization. Options are all very, very close, but you have to think, if you are doing IVF, why are you doing IVF? Mostly when we do IVF, there are two reasons. One is either the male sperm is not able to fertilize the ovum. So we do IVF, we do it outside. Sometimes female system also is not able to conceive, it's not able to undergo fertilization. So we do that fertilization outside, and then we do what? So what do we transfer? And look at where they are asking. So always if in the same question they ask you transfer of which of the following into the uterus, option will be different. But in IVF we do transfers in the fallopian tube. So what do we transfer in the fallopian tube? What is possible to transfer? A, embryo only up to 8 cell stage. Is it only embryo that we can transfer?
No.
No. So I think option A is outright wrong, right?
Yes, sir.
Option B, either zygote or early embryo up to 8 cell stage. Then embryo of 32 cell stage, and D is zygote only. So what do you think is the right answer among the women?
Option B.
B, up to
Yes,
8
sir.
only, like we cannot go beyond 8 cell stage.
Yes, sir. For fallopian tube, we cannot go beyond 8 cells.
Yes, right. We can, in like technically we can transfer anything from a zygote to a embryo of like even 64 cell stage. But the point is, where are we transferring it will. So if we are transferring it in fallopian tube, it could be either zygote, or early embryo, because if it is a later embryo, it may have chances of getting implanted. The later the embryo, it gets implanted, right? Remember? So we have to, if we are transferring it into the fallopian tube, we have to give it enough time to cross the fallopian tube and reach the uterus to get implanted. So the correct answer is B. Right, Rifat? Yes, correct. Others also understand why it is the correct answer?
Yes, sir.
Okay. Question number 15, Atif. We'll do some questions here and then we'll start with genetics. Question 15.
Okay, sir. Artificial insemination means artificial introduction of sperms of a healthy donor into the vagina, introduction of sperms of a healthy donor directly into the ovary, transfer of sperms of a healthy donor to a test tube containing ova, transfer of sperms of husband to a test tube containing ova.
Artificial insemination. Rifat, which one? Which option?
A.
Others?
Option A.
A, right. Others, artificial. See, if it is insemination, it cannot be outside the body. That's the only logic. Okay. Otherwise, it is in vitro fertilization, right? So if it is saying insemination, then it has to be inside the woman's body. And if it is inside the woman's body, then what is the point of directly putting sperm into the ovary? It never goes into the ovary. So the only two options are A or B, and A is the correct one because you introduce the healthy sperms into the vagina. That's called artificial insemination. Okay. Now question 16. I'll read and Hanifa will give me the answer. One of the legal methods of birth control is A, by having coitus at the time of daybreak, by a premature ejaculation during coitus, C, abortion by taking an appropriate medicine, or D, by abstaining from coitus from day 10 to 17 of the menstrual cycle. Which one do you think is a legal method of birth control here, Hanifa?
B.
Sorry?
So B, by premature ejaculation during.
Is A, okay, let me go to Atif. Atif?
Sir, option D.
Okay. You all are falling in the trap actually. Anifa. Rifat.
Sir, I'm changing to option C.
Why are you changing to option C now? Because I gave you a hint that you are falling into the trap, and this is the only trap. See, what is illegal? Is abortion illegal? Or is
Sir,
determination of the sex during fetal uh, development is illegal? Illegal is knowing of the sex and then aborting it, right? You
yes, sir.
understand? Option C is simply, in other twisted words, an alternate sentence for taking a pill, a contraceptive pill. When you take a contraceptive pill, when females take a contraceptive pill in emergency, what are they basically doing? They are taking an appropriate medicine that aborts whatever is inside the uterus, correct? Yes or no? So they're just tricking you. A, B, and D are not foolproof birth control methods. You understand? By having
Yes,
coitus
sir.
at the time of daybreak, this does not make sense, right? Daybreak. By a premature ejaculation during coitus. What is a premature ejaculation? There is nothing like there is an ejaculation or no ejaculation. No premature ejaculation. A and B are incorrect options. They are not options at all. D, by abstaining from coitus from day 10 to day 17 of the menstrual cycle. It is a method. It is, but it's not a method of birth control. It's a method to not get pregnant in the first place. If this does not work, there will be a fertilization and there will be a embryo developing. So the only workable method here of birth control is abortion by taking an appropriate medicine. Now, by putting the word abortion, they are making you think that it must be illegal. But remember, abortion after amniocentesis for a specific sex is illegal, or abortion after a particular period is illegal because it is harmful for the female after the first trimester. But before that, if someone is pregnant and wants to get rid of the baby, they can do it with their consent, with parents' consent, through pills or MTP kits. In MTP kits also there are medicines that they which gives all the everything out. Okay, so C is the right answer. Everyone understands? Hanifa, Rifat, Hiba, you have to read the option carefully before answering. Okay, Atif?
Yes, sir.
Yeah. Okay, so I think we discussed quite some questions, some confusing, some very easy ones as well. So let's start with today's new chapter that I'm going to begin, which is genetics. Everyone back? Do people need 2 minutes of break? Let me get some water for myself. You also get water, okay? Then we'll start with the fifth chapter. Okay? Atif,
Okay, sir.
just 2 minutes. I'll be back. Okay, hello everyone. Am I audible?
Yes.
Okay, cool.
Yes, sir.
Everyone is back. Hanifa, Rifat, Hiba.
Yes, sir.
Okay, so let's start with chapter 5, which is we are starting genetics from here onwards. Before I start with genetics, let me ask you some basic fundamentals that you have already covered in your junior classes. You all have studied about Mendel, right?
Yes, sir.
What did Mendel do? Mendel did some experiments, correct?
Yes.
What was the experiment about? Anyone can brief me about it? What were Mendel's
He
experiments?
crossed green peas.
Hmm? Green peas. Yeah. Are there other peas as well? So you are right. He chose pea plant, right? The
Yes.
model system he chose was pea plant. Anyone remembers the scientific name of pea plant? Because in entrances they don't give you the common name sometimes. It's Pisum sativa. Okay, Pisum sativa or Pisum sativa. So he chose pea plant and did some experiments. Can you tell me what did he do basically? He crossed pea plants and then what? Why was he crossing pea plants? Atif. Since you talked about it that he crossed pea plants, why was he crossing?
He did monohybrid cross with pea plants. I think tall and short pea plants. I'm not sure. And then he found out that I think 3 of them are tall and 1 of them are short. Like, yeah.
Yeah, so you are right. He actually was looking at some traits of the plants, right? So one thing that is clear from Atif's answer is that he was crossing between plants, and then he was looking that if he crosses a plant with some trait and with a different plant with different trait, what happens to the progenies? What trait do they get? Okay, right? So before going into that, let's cover why did Mendel chose pea plant? Why not any other model system? So write down a question. This can come in the boards. Why Mendel chose pea plant? The answer to this is he chose pea plant due to the following reasons. Due to the following reasons. First reason is pea plant had many traits, contrasting traits that could be studied. So it has contrasting traits that could be studied easily. Okay, so whether they are going into the next generation or not can be studied easily. Next, if you are doing a genetics experiment, let's think it like that. Okay, and okay, Rifat and Atif, if you two are doing an experiment of genetics and you're looking at some traits in the next generation, so first thing that whatever model system you choose, there should be traits in it to look for, right? If you take something which do not have traits, contrasting traits, then you will not be able to tell, but just by looking at the organism, whether inheritance or genetics has happened or inheritance is happening or not, right? So contrasting traits has to be there, correct? What apart from that you need in your model system? You need a short lifespan, yes or no?
Yes.
Yeah. And pea plant has a short lifespan. So write down short life cycle. Sorry. We are mostly interested in cycle here, short life cycle. It's around 3 months for pea plant. And short life cycle traits that we can see. And we should also get many progenies, right? If we just get 1 or 2 progeny in the next generation, then will it be of use to us? Right? We should get hundreds and hundreds of progenies easily. So write down pea plant produces many progenies, large number of progenies. Basically, by progenies, we are talking about seeds, right? Seeds are the progenies, yes or no?
Yes, sir.
Because we can then sow the seeds and look for the traits in the next generation. And anything else? If you are doing an
Easily available.
yes, one thing is easily available, but if you are doing an experiment on organism and you are breeding it, availability is not a problem because you are breeding it. But something related to your answer, easily available, it should we should be able to easily do both, either cross fertilization or self fertilization. Remember, in plants we studied cross pollination, self pollination,
Yes, sir.
and we
They
also
have bisexual flowers.
yes, and no, we also studied that pea produces cleistogamous flowers. Remember, Rifat?
Yes.
He was an example of cleistogamous. So autogamy is already going to happen, and if I want uh, deliberately to do cross pollination, then there are certain techniques in which you just open the flower, cut the anthers, or remove the female part, and then just allow cross pollination to happen. So that was easily. So both, right on, both self pollination and cross pollination was possible. Plant was compatible for both. That's why Mendel chose pea plant. Now, when he chose pea plant, what are the characters that he studied? And those characters are important. Mendel studied around, write down another question. What were the characters? What were the characters that Mendel studied in the pea plant? Okay, tell me which ones do you know? Which characters do you know Mendel studied?
Shape.
Okay, before going to that, tell me what is the difference between um, character, and then there was, you remember studying dominant and recessive trait?
Yes, sir.
Dominant trait and recessive trait. What was dominant and recessive, Atif?
Like dominant trait means like the dominant trait means that if the trait if it is the trait if it is both heterozygous and homozygous, then in both cases dominant trait will be expressed, and recessive trait it has to be homozygous for it to be expressed.
Okay, yeah, that's good. That's right. But to understand it better, if I ask you, what is the difference between a character and a trait? Character and a trait?
Sir, I'm not sure. I think character is like a category, for example, eye color, height, etc. And then trait is like the different types of that single character. Like for eye color, we have like brown, blue, etc. And for height, you have tall, short. Yeah, I think like that.
Yes, you are perfectly correct. So the character is like height. So one of the character that Mendel also studied was plant height, remember? Or the stem height, with which Atif also started his answer. So for the stem height, do you know what was the dominant trait and what was the recessive trait?
Tall is dominant and short is resistant.
Yes, or I can call it dwarf, right? Or short is also not incorrect. It's fine. So stem height was one character. Then what was other characters that he studied? Seed. Someone said seed color, right? For seed color, and with seed there was another character he studied, the seed shape. Now, can you tell me for seed color and seed shape what were the dominant and recessive traits, if you remember? Seed color.
Yellow and green.
Yellow was the dominant trait, seed color.
Yes.
Okay. And recessive was
Green.
green. How many of you have peas in your home, coming in your home, or you like peas? You have seen the peas that come to your house? What is the
Yes.
color of the seed of the pea, which you put in vegetables or pulao or whatever?
Green.
Green, no? Do you ever see yellow peas coming to your house?
No, sir, not me.
Why? Then do you think that we are deliberately propagating a recessive trait because we just like it? If we leave the pea plant in wild, the dominant trait is yellow, right?
Yes.
So most of the peas must be yellow, but we are choosing only that pea which is green, which has this recessive trait of green seed color, and we are propagating it further. That's why we do not get yellow peas at home. Now talking about seed shape, what was the dominant trait about seed shape?
Round and wrinkled.
So round was dominant and wrinkled was recessive. Am I correct?
Yes.
So for our purpose, we are propagating one round, one dominant character with one recessive character. Yes. We do not know about the stem height because we never go to the farm and see the seed, see the pea plant ourselves. But talking about seed color and seed shape, we know that in our house we get a combination of a dominant and a recessive trait. Yes or no? They are round, but they are green. Am I correct, people?
The difference between yellow and green peas is only the color, right? Is there like any specific difference?
Sorry, what? Difference between
Difference between yellow and green peas, is there like any
color? Yeah.
specific? Is it only the color, or is there any specific difference?
No, it's the color and the pigment that gives that color. That's all.
Then
Like
why are green peas like more preferred?
probably because of that pigment, there might be some taste. Like green seeds of the pea are mostly little sweet, on the sweeter end, and that is one reason I can think of. Like, there has been in the market, people must have preferred green. That's why green started getting propagated. As per the nutritional value, I'm not sure, or I don't think that there is a difference that green peas are more nutritious or something. It's the same for both. Okay.
Yes, sir.
Now, apart from seed, what was the other traits, other characters that he studied? Any idea? He also studied the pod in which the seeds come, right? So there was similar thing about the pod color and pod shape, remember? You know, you all know pod, right? Which we open and then seeds from the inside we take out.
Yes, sir.
Yeah. So what is the dominant and recessive pod color? Any idea? The dominant pod color is green. You can remember it to be opposite of the seed. Seed color dominant is yellow, so pod color dominant is green and recessive is yellow. Whereas pod shape, yes, pod shape is just like the seed only. It has two, either a full or in some books it's written as inflated, and the recessive is constricted. So the difference between this is, this looks like an inflated pod in which you have seeds, right? And here you will see that if there are seeds, then the pod is just, it takes the shape of the seeds. It's not inflated. Is it clear? So this is
Yes, sir.
full and this is constricted pod, which is a recessive trait. And that's all. Apart from that, yes, there was one more important one, flower. Flower color. Anyone remember the dominant color of the flower? In case
Violet.
yes, they have violet and they have white flowers. So violet was the dominant one and the recessive one was white. We're going to use all this information. That's why I take some time there. And apart from that, I think one last was flower position. Where did the flower appear? Now with flower position, I mean there are two possibilities. One is where, say, this is the plant, okay, and as it grows, on each alternate branch there are flowers emerging. So the plant keeps growing and flowers keep emerging at, this is known as axial arrangement, okay, because on one axis or the other axis flowers are emerging. Is it clear, everyone? The
Yes, sir.
other is where the plant has the flowers right at the terminal end, like where it ends. So if it has three branches, at the end of all the three branches, there will be flower. So this is called terminal. Terminal means where it ends. So for flower position, the dominant trait was axial, because if it was not axial, then it will not be a tall plant. You understand, everyone, the correlation between the two? If flowers are axial, then the plant will keep growing. You understand?
Yes.
If the flowers are terminal, then it will be a dwarf, more likely. So write down all these characters and their dominants and recessive traits. Written?
Yes, sir. I wrote.
Others? Shall I move forward?
Yes, sir.
Okay, before I move forward, I want all of you to be sure about, I think we have discussed this. So you all are clear about these terms. Chromosome. Everyone knows what is a chromosome? Rifat, do you know what is a chromosome?
Yes.
Yes, can you tell me? Then we'll move forward if this concept is clear. Anyone who does not who's not clear with these terms, please ask me. Because if you don't ask me, I'll move forward and I'll assume that you know this. We have discussed this before, so that's why I'm asking. What is chromosome, Rifat?
It's
Hmm?
it's made up of DNA.
Yes, it's a super coiled DNA. Remember?
Yes.
It's a DNA wound on proteins and supercoiled to be kept in a very small space. That's called a chromosome, correct? Just like a thread on a spool. I gave you the analogy also. Remember? So chromosome has two things, DNA and protein, right? Everyone, chromosome is clear, right? And genes are inherited through gametes in the form of chromosomes. We get one copy of chromosome from the mother and one copy of the chromosome from the father. Yes, everyone clear?
Yes.
Very good. Now, you all know what is gene? Everyone is clear about gene? Atif, this question is to you. You know what is gene?
Gene is like part of the DNA,
Yes,
which like has code for a specific trait. I
yes,
think.
yes, right. So it's a actual physical region on the DNA itself. It is DNA itself, right? It has a starting point. Remember, start codon, stop codon. We studied that, right? We did that. Yes or no?
So are you talking about last year?
I'm talking about like this year only. In the beginning, we talked about DNA, gene, the initial chapters. Yes. Don't remember? I told you that gene has a beginning. Every gene on a DNA has a start codon and a stop codon, and in between is the open reading frame.
Yes, and now I remember.
Remember? That was a gene, okay? Good. How many of you also are clear with the term alleles? What are alleles? If not, we will discuss. Hanifa, allele? You know what is an allele? Or what are allele? Yes or no? Hanifa, you're not audible to me if you are saying something.
No, sir.
No, you are not clear. Okay. Rifat, you know what are alleles?
No, sir.
Okay, so let's discuss this. So you are clear about chromosome, DNA, and gene. Very good. Now let's go up here. You know that this character, which is let's say height. For every character, there must be a gene, right? A gene gives rise to a character. Yes, one or
Yes.
multiple genes giving rise to characters. Now, for every gene, there are versions. It's not like all the genes present, like all the copies of the genes are exactly the same. So they are called alternate versions or other forms of the same gene. For example, what gives rise to tall is capital T, right? And what gives rise to short is small t, that you have studied in previous classes.
Yes, sir.
So this gene copy here, which is slightly different from this one. So this gene has two traits. One is a dominant trait and one is a recessive trait. So this dominant trait is coming because of a allele, and this is that allele. And this recessive trait is showing up because of a other version, another copy of the gene, which is this allele. So write down, define allele. Alleles are alternate versions of the same gene. giving rise to contrasting traits.
So gene is for character and allele is for trait, right?
You can say that actually, because ultimately allele is a genome. Allele is also a gene, but I'm saying it allele because on a chromosome, let's say this is chromosome number 1 of mother and this is chromosome number 1 of father. Both are chromosome number 1, okay? 1 and 1. And on this chromosome, there is a gene located here. So in both, it will be located at the same place, right? Let's say this is the
Yes.
gene for hair color. Now mother, the allele that mother gave is, the gene is for hair color, but this particular allele here is about brown. It gives brown color and curly texture to it. Whereas this allele of the same gene, the gene is for hair color. Let's say it gives for black hair color. You understand? Let's only talk about color now, not the text. So you can understand that the genes are the same. It's the gene for hair color, but the allele that mother brought in was for brown hair, and the allele that father brought in was for black hair. Is it clear? Do you understand, everyone? But these two pairs present at the same place are known as alleles. Alleles are always present at the same location in uh, in the homologous chromosomes. Is it clear? Do you understand, Atif?
So what are homologous chromosomes?
One coming from the father, pair, like pair chromosomes. One from the father, one from the mother, making a diploid set.
Okay.
They are always present at the same location. Okay, everyone understands?
Yes, sir.
Yep. Okay. So if this concept is clear, then let me go directly to discuss Mendel laws. You have also studied about Mendel's laws of inheritance. At least you have studied the name. Do you remember the name, anyone? Mendel gave
Law of segregation.
the first law you remember is actually the second law. So remember there are three laws. Anyone remember the names of the three?
I think the first one is law of independent factors.
No.
Then law of segregation. Then the final one.
Final one is law of independent assortment. Like final
Yes,
one,
sir. That one.
final means it is the most complex one. So that's why we call it the final one, which is the most simplest one that we have just discussed above. Capital D, small d. First was called law of dominance. Remember?
Yes.
Law of dominance. Remember, everyone? What does the law tells you? Write down. Let's state the law first. Law of dominance states. Law of dominance states yes, you can see this, right? Write down. Law of dominance states that when present in a heterozygous condition, when present in a heterozygous condition, one allele Mendel at that time did not know them as alleles, so he used the term factors. Okay, so you can write in brackets factors. One allele or factor is dominant over the other. And always express in the presence of the recessive allele. And always express itself in the presence of the recessive allele, which remains hidden or masked. The condition I'm talking about is this: if there is a capital T and small t present in any seed, which means that one allele came from the father, let's say, and the other came from the mother, right? Yes or no? So in this condition, this one allele is always dominant over this recessive one. Is it clear, everyone?
Yes.
And this will always express itself in the presence of recessive, and recessive will always be masked. So the only way recessive can express is in a homozygous condition. So write down this second line. A recessive trait, according to this law, according to this law, a recessive trait can only express itself in a homozygous allelic condition. In a homozygous allelic condition or homozygous allele pair. Okay. So by this law,
So in a homozygous
allelic condition, if it is capital T, it is also tall, right?
yes, sir.
If it is capital T small t, this is also tall, right?
Yes.
The only possibility of a short plant is this. This will be a short or a dwarf plant, right? So
Yes.
what is the difference between first and second? Both are tall, but this is called a homozygous tall. Okay, everyone? Or we call it a pure line tall. Is it clear?
Yes.
And this is called heterozygous tall or a hybrid tall. Is it clear? Short or recessive trait is always homozygous. So homozygous or pure line recessive.
Sir, is there any specific reason like why one allele is dominant or the other is recessive, or it's just the way it is?
Yes, there is a reason. I'll tell you about the reason, but let's first understand the concept. Okay, for every gene, the reason might not be the same, but the overall reason is that one type of whatever that gene is doing. So mostly genes produce mRNA, right? DNA produces RNA and RNA produces protein. Remember, I told you this?
Yes.
So all genes, all functional genes produce a protein, right? That protein can have any different function. Some proteins are receptor proteins, some proteins are, let's say, enzymes, some proteins are something else, some are pigments, like in case of green and yellow colored, that is the pigment. So whatever function that protein is playing, in evolution, some of the alleles do better than the other for the survival, maintenance, sustenance, and adaptation of that animal. Okay. So whatever trait is more beneficial to an organism in evolution, that becomes the dominant one over time. Because if that expresses, the organism has more and better chances of survival in evolution. So that is one reason. It is not the only reason. Okay. Other, yeah. So other kind of reasons are that because of some mutation, one becomes dominant and the other becomes recessive, not stable. So recessive proteins, if a protein is not stable, like at that given particular environmental condition, like say temperature factor, or in that region, if that protein is not stable, then obviously it will not sustain, right, in the presence of a more stable protein. Yes or no? So the gene that codes for that more stable protein will become the dominant gene automatically. That is another, that could be another reason. You understand?
Yes, sir.
Yeah. But it happens that most of the genes which have multiple alleles, one allele is dominant over the others. That's always seen. Okay. So in this heterozygous condition, we get to know about the Mendel's law of dominance. Is it clear?
Yes, sir.
And what is a dominant trait and what is a recessive trait? So the definition for dominant trait is that dominant traits always express itself, whether in a homozygous, write down, a dominant trait always expresses itself whether in a homozygous condition or a heterozygous condition. Dominant trait always express itself whether in a homozygous condition or a heterozygous condition. Okay. Whereas a recessive trait only expresses itself in a homozygous condition. This is why in NCERT and in other books it is said that recessive trait is that trait which is not expressed in the F1 generation and is expressed in the F2 generation, right? This you must have read. Yes or no? So in
Can you repeat what you said?
the recessive trait remains hidden in the first generation and it shows up in the next generation. So a recessive trait is that trait which can stay hidden. It is there, it is present, but it stays hidden. Now, why it is said that it stays hidden, you have to not just remember, you have to also understand. It is always hidden if we start with, so let me start a cross here. Let's suppose if I start a cross, let's suppose if I start a cross here with a pure line tall, capital T, and I cross it with a pure line short, small t. Okay. If this is one parent and this is the other parent, and I'm making a cross between the two. So you know how to make crosses, everyone? We make crosses
Yes,
with Punnett square. You know Punnett square?
yes.
Yeah. So basically there are three simple rules. Write down. To make crosses through a Punnett square, there are three rules. First is write the genotype of the parents. So this generation becomes the parent generation. And these are the genotypes. So we first have to write the genotype of the parents, first step. Second, find out number of gametes that can be produced from each parent. Number of gametes that can be produced from each parent. Now in this case, it's capital T and capital T. So when We are, and what we are studying here is monohybrid cross. This is called a monohybrid cross, or also known as single gene inheritance. We are talking about just one gene here to understand law of dominance. Okay. So on one side, we will write, let's say this is the male, and let's say this is the female. So we will write we are supposing, so assuming that this is the male, so we are writing the gametes of the male. So it is a diploid setup. You can see capital T, capital T. There are two pairs. So when they will be segregated in gametes, it will be T and T, right? Yes or no?
Yes.
Same on the other side. It will be small t and small t. Now, can you tell me if this is the parent in F1 generation? We call it F1 because F stands for filial, and filial means generation. So filial 1, generation 1. Okay. So in F1, what will be the genotype of possible 4 progenies? Capital T, small t, one from this side and one from this side, right? Similarly, capital T, small t. Here also capital T, small t and capital T, small t. Yes or no? Everyone, do you understand? How are we making it?
Yes.
Hanifa, is it understood to you as well? Rifat, Hiba. Say yes or no, or write at least yes or no, because this is the part, if you do not understand, there will be confusion afterwards. Because after this, I'm going to talk about 2 gene interaction. Hanifa, is it clear?
Yes, sir.
Good. Now see, this thing that we got a new combination of a genotype. This is called a genotype. A genotype is what is the makeup, genetic makeup of the allele. So all the 4 kids, all the 4 offsprings got a hybrid setup, right? And phenotype is that all of them will be tall, right? Phenotype is what we can see. Phenology is the study of visible characters. Phenotype is what is the visible, what is the character outcome of the genotype. So all will be tall, but what kind of tall all of them will be? Pure line or hybrid?
Hybrid.
Yes, so all will be hybrid tall. And this is the genotype. Is it clear?
So
Sorry.
what's the difference between phenology and morphology?
Sorry. This is the phenotype. Yes, Atif, I'll come back to the question. But you understand this is the phenotype
Yes, sir.
and this is the genotype. Yes, what are you asking?
What's the difference between phenology and morphology?
Phenology, when we talk about, see, morphology is anatomy and morphology goes hand in hand, where anatomy is the study of internal structures, morphology is the study of outer structures, like the structure which are outside. Okay, that's morphology of that structure. Like if you see the morphology of a flower, you can dissect a flower and can see the structure as well. When we talk about phenology, we are only talking about some characters, some visible characters that are different from one organism to the other. So phenotype, morphology and phenology are like, morphology is the study of outer structures and phenology is the study of visible characters. So those characters may or may not be structures. For example, uh, uh, height. Height is not a structure, right? It is a character. You understand? Skin color, it's not a structure, it's a character. But flower shape is a structure. You understand? So morpho is all about structure. What is the structure of that flower? Is it elongated, bell shaped? It is round? Is it like it has 10 petals or 5 petals, or is it like a rosette? What is that structure? But phenology is about the height, like height. If you see two plants, one tall, one short, it's just like this, both the plants look the same, one is longer, right? Yes or no?
Yes, sir.
So if I tell you there are two pencils, one is this and the other is this. So you can clearly see that the both have similar structure, right? Morphologically, they both are same. Yes or no? Both have a back
Yes, sir.
and a point. But phenology wise, when we will study that, okay, one was short and one was tall, because we are also taking into consideration characters here. In morphology, we might not take character, which is not structurally uh, analyzable. Is it clear?
Yes, sir. It's
Yep. So phenotype, genotype is clear to you. Now let me ask you a question. If we start with pure line parents, that is what you have to always understand. NCRT always talks about parents when they are pure line. So if parents are not pure line, then in F1 generation, this will change. Okay. So the F1 generation depends on what was the genotype of the parents. So if parents are pure line, one is dominant pure line, which is tall, one is short. So when I crossed a tall parent with a short parent, in F1, I got all the tall kids, right? All the tall plants.
yes.
Understand? Where did the short gene went? It became, it is always present as a heterozygous allele with the dominant allele, and it is masked, it is hidden. You understand? So it is not showing up in F1. Next question. Is the F1 tall same like the parent tall? Atif? Is the F1 tall which we get, do you think it is the same as the parents tall?
Yes, sir.
Genotypically no, phenotypically yes. So if you see phenotype, both are tall, they both will have the same height. But if you see the genotype, one was pure line or a homozygous tall and the other was a heterozygous or a hybrid tall. Do you understand? So whenever this question is asked, it is like the same tall, you have to say no. Genotypically, they are not the same tall. Okay? But phenotypically, they both are tall. Okay, everyone? They both will look tall, but their genes will be different, because tall can also come up in a hybrid state. Tall can also express in a homozygous state, following the law of dominance. This is law of dominance. Now, if I go one step ahead and I self cross F1, if we are doing self cross of F1, which means we are capital T, small t, capital T, small t, we are doing this cross. Can now all of you do it and tell me what will be the genotype and phenotype ratio in F2? Do it please quickly. You have to tell me genotypic ratio and phenotypic ratio. What was the ratio here? There was no ratio, it was just one. All had the same genotype and all had the same phenotype, correct? No ratio was there.
Yes.
Now tell me, do this cross and tell me quickly.
So the phenotype ratio is 3:1 and genotype ratio is 1:2:1.
Okay. Now let's follow the steps. First, we'll make a Punnett square. Everyone did the same.
Yes, sir.
What should I write here? Atif, please lead me. On one side, what will be the two possible gametes?
Capital T and small t.
Okay. These traits will go in each T gamete. And on this side,
Capital T and small t.
yes. And what will be the progenies? This one?
Capital T, capital T.
Yes. And this one?
Capital T, small t.
This one?
Capital T, small t.
This one?
Small t, small t.
Now, what will be the genotypic ratio? How many types of genotypes are present? One I can see is this capital T is one type of genotype, right? Other genotype present is this capital T small t, correct? And anything else that can be present? Small t, right? And what is the ratio for these three possible genotypes? 1 is to 2 is to 1. Yes? Everyone understands the genotypic
Yes.
ratio?
Yes, sir.
And what will be the phenotypic ratio? Phenotypic, because there is law of dominance, there can be either two traits, tall or short. So simple, if you are following law of dominance, either a plant can be tall or a plant can be short, right? So what will be the phenotypic ratio? How many of them are tall? Is this tall? Hanifa? Is this plant tall? Capital T. Yes. Are these two tall? Atif? Atif, you're there? Atif is not speaking. These two are also tall. And this is the only short. So we get a ratio between tall and short as 3:1. And in this 3, these two are hybrid tall. But whether it is a hybrid tall or a pure tall, we cannot tell it just by the appearance. Can we? Can we say it by just by the appearance? No. Am I audible to everyone?
Yes,
Rifat, am I audible?
yes.
So you understand the genotypic and phenotypic ratio? Everyone understands? So this is where this is the ratio in F2. F2 ratios. Now this F2 ratio is such when we started the parents as pure line. And these are the parents. Then we self cross the F1 and got F2. Is it clear, everyone? If it's clear,
Yes,
I
sir.
will give you all a homework, like just a one to do exercise. Write down the question. Now, the simple concept that you just understood will not be asked very simply, like in this manner, in your exams. It will be asked in a complicated manner. How? Write down the question. Uh, a homozygous tall plant, was crossed with was crossed with a heterozygous tall plant. Heterozygous tall plant. What will be the genotypic and phenotypic ratio in the next generation? Now, to solve this, you have to consider them as parents. You understand, everyone? You understand? Atif, Rifat, Hanifa, Hiba? Yes.
Yes, sir.
Please solve it and write done when you are done in the chat, okay? Take 2 minutes. Done, people? No one has written done. Those who have done, please write done. I've just twisted the question by changing parents. Now you don't know what generation is this, so just assume them as parents. They are being crossed, so the next generation will be the F1. First done.
Yes, sir.
Done. Okay. That is also done. Iba, all of you are understanding, right? Anyone who's not understanding? Okay. Yes, sure. Iba, I'll be waiting for you, okay? Please let me know when you're done. Okay, so Rifat, will you lead me towards solving this? Let's solve this. Rifat, you're there? Yes. So first, tell me this parent 1. What do you think is the genotype of the parent 1?
Capital T and capital T.
Just one moment. So we'll add the genotype for it. It will be homozygous tall. So it will be capital T and capital T, right? Yes. And for this parent?
Capital T and small t.
First step, write the genotypes of the parents that we have done. Now we have to go to making the Punnett square. Tell me, on this side there will be capital T, and this side there will be
Capital T and small t.
capital T small. Now can you tell me what is the genotype of each?
Capital T and capital T. Capital T and small t.
Okay.
Capital T and capital T.
Okay.
Capital T and small t.
So what will be the genotypic ratio?
There are no genotypic is 2:2.
Sorry,
2:2.
2:2, right?
Yes.
And what are the genotypes? Capital T is one genotype and capital T small t is another genotype. So it will be 2:2, correct, everyone? What will be the phenotypic ratio? It will just be 1 because all will be tall, right?
Yes.
No ratio, just 1 because all will be tall. But two of them, these two, you know, are pure tall. Correct?
Yes.
Like this parent. And other two are hybrid tall, like the other parent. You understand? So
Yes.
the progenies resemble their parents. There is no third different genotype present here. Understand, everyone?
Yes.
Yeah. Now let's do one more question quickly. Write down the question. Uh, a heterozygous tall a heterozygous has to be tall, right? If it is following law of dominance, because a heterozygous cannot be short. But still, I'm telling you, a heterozygous tall is crossed with a short plant. Now, tell me the genotypic and phenotypic ratio in the next generation. Time starts now. Now do it fast. And this time when you will answer, you will write the genotypic and phenotypic ratio in the answers in the chat. Okay, everyone? Give me the answers in the chat and as quick as you can. Okay. Hiba and Hanifa. I'm waiting for your answers. Hanifa, genotypic, not genetic. Genotypic, okay? Genotypic and phenotypic. Okay. Hiba, are you there? Am I audible to you? Will you please confirm if I'm audible?
Yes, sir.
Yes. Are you having some issues writing the answers in the chat, Hiba? Okay, so I will ask Atif to lead me this time. Atif, you're there?
Yes, sir.
Yes, let's solve this question. So tell me first about this parent's genotype. What it will be?
The genotypic ratio.
Just the genotype of parent A, this parent.
Okay, sir. It will be capital T, small t.
Capital T, small t. Okay. And for this one,
Small t, small t.
let's say this is parent B, this is small t, right? So we are basically doing this cross. Am I correct, Atif?
Yes, sir.
So let's make it. What will be on this side?
Capital T, small t.
Okay. Here,
Small t, small t.
right? Okay. Tell me what will be the first offspring?
Capital T, small t.
Okay. Next one.
Capital T, small t.
Okay. This one.
Small t, small t.
And
Small t, small t.
okay. Now what will be the genotypic ratio? Atif?
Yes, sir. It's 2:2.
2:2. What are the genotypes? One is 2 is capital T small t and 2 is small t, right?
Yes, sir.
And what will be the phenotypic ratio?
2:2.
Yes, 2 tall and 2 short, right?
Yes, sir.
And the tall in this case are hybrid tall. So the question why I'm repeating this all the time, because the third question that is asked in entrances is, if I'm so the listen to the question. It is the same question that you just solved, but they will not ask you genotypic ratio or phenotypic ratio. What they will ask you is, if a heterozygous plant is crossed with a short plant, okay, then in the next generation, the tall progenies will be option 1, genotypically same as parent, genotypically different from parent, third will be both same and different from parent, and fourth will be can't say. What will be the answer? The question
Can you
is,
repeat the question?
write down the question, everyone. Write down this is how they are asked. This is asked in entrances. If a heterozygous plant is crossed with, if a heterozygous plant is crossed with a short plant, then in the next generation, the tall progenies, obtained will be, option 1, option A, genotypically same as the parent plant, genotypically same as the tall parent plant, option B, genotypically different from the tall parent plant, option C, both same and different genotype will be produced, both different and same genotype will be produced, and option D, can't say about genotype in F1, in the next generation, in F1 generation. Can't say about the genotype in the F1 generation. Atif, what will be the right answer? Atif, is it confusing? Am I audible, people?
Well, I'm having some problems. Yeah, I'm really sorry, but can you repeat the question once more?
Yes. Anyone has written, like, you could not write the question, the whole question?
Yes.
The question is, if a heterozygous plant is crossed with a short plant, then in the next generation, the tall progenies obtained will be,
The tall progenies?
the tall progenies obtained will be which of the following? Will be which of the following? And there are 4 options. First option is genotypically same as the tall parent plant, genotypically different from the tall parent plant, both same and different progenies will be produced, or fourth, can't say. Could you understand, Atif?
Yes.
Yes. So what do you think is the answer?
First one.
Yes, right? Because see, this tall that you get, capital T small t, is a hybrid tall, right?
Yes.
And the parent was also a hybrid tall to begin with, right? So in this case, tall is just like the parent plant. Yes.
Yes, sir.
If we would have gotten this kind of tall in the progenies, then the right option would be different, right? And if
Yes,
we
sir.
would have gotten both, then the right option would be C. Both same and different genotypes are produced from the parent. Everyone understands?
Yes, sir.
Yep. Okay. So this is law of dominance. Is it clear to all of you?
Yes, sir.
Now in the next class, write down last 2 lines. Law of dominance is not a universal law. Law of dominance is not a universal law, which means it is not true always, is not universally accepted. It has exceptions. It has exceptions. And tomorrow we'll start with these exceptions. So your homework is you have to go and read about two things. First is incomplete dominance, which is the first exception of the law. When Mendel gave this law, he did not know about this. And second is codominance. When you will study codominance, incomplete dominance I will teach in Snapdragon flower. Snapdragon flower color. That you have to study. And codominance is there in ABO blood group, human blood group, okay? Just study how the blood groups are decided and things that we will do in the next class. Is it clear, everyone?
Yes.
These are the exceptions for law of dominance that we will do in the next class. Okay. In the next class, again, we will meet previous year's questions from last and third chapter also. Okay. So keep revising what we have done previously as well. We will do questions from this topic once we have finished a major chunk. Okay. Okay. Anyone has doubts regarding today's class? Anything you want to ask? Anifa, Rifat,